Hello, everyone, and thank you for joining us today. My name is Dalton Dietrich, and this is our opportunity to talk with various scientists within the MIND project. Really excited today that we have Dr. Hassan Al-Ali, who's assistant professor in the Department of Neurological Surgery and an extremely important member of the MIND project to cure paralysis. Hi, Hassan, how are you doing today?
Hey, Dalton, I'm doing well. It's good to see you.
Good to see you, thank you. Well, I've been very excited since Hassan has joined the MIND project faculty because of the fact that he brought with him these new technologies in developing new drugs, a new discovery platform, if you will. So first, Hassan, why are we very interested in developing new drugs and new compounds for spinal cord injury?
Great question, Dalton. Decades of research have shown us that the problem of regenerating the central nervous system is a very complex one. So, we've revealed a lot of the underlying biology and a lot of the mechanisms that sort of synergize to prevent the growth of these axons and the repair of neural networks once they're damaged. So what has become obvious over time is that tackling one mechanism of failure to regenerate is not yielding sufficient regrowth of the axons to have a clinically meaningful recovery. So what we at the M.S.M project are doing, and our group specifically, is trying to understand what kind of paradigm shifts are required in the way we think about drug discovery and drug development so that we can bring something to the patients, something to the clinic that is efficacious, that's going to produce the kind of histological and physiological recovery, but also functional recovery and improvement of the quality of life for the patients.
Yes, you mentioned the complexity of spinal cord injury, and there's a lot of interesting targets at the cellular and molecular levels that scientists all over the world are trying to look at in terms of enhancing or depressing those pathomechanisms. But at the end of the day, scientists feel that actually repairing the nervous system by axonal regeneration is the target that's going to most likely make the most difference in people living with spinal cord injury. So you've brought some new technologies to allow you to identify novel drugs, compounds, and then to put those into more translational models to test to see if they're going to actually make a difference in terms of functional recovery.
Right. So that's absolutely true. And there's a pain in the pharmaceutical industry in figuring out how to identify the target that you're going to develop your therapeutic against, right? So that's the first part of the drug development campaign, and arguably the most important part, because if you don't know what your drug is going to target, it's kind of like walking blind, right? So the molecular biology revolution and the sequencing of the genome has brought us tremendous amount of information and technologies to help us understand the workings of the cell and the pathways that are involved in, as you said, regulating the growth of these axons, promoting them or suppressing them. But a pain that has remained in the field is, how do you go from integrating all that information, and how do you go from understanding what are the pathways that are involved to figuring out specifically what is the gene or gene product that your drug needs to target to be efficacious, to be safe, to give you the clinical outcome that you want without causing any side effects, as few as possible, to the patient, right? So our group, and which is led by me and my collaborators, John Bixby and Van Slammen, we're always trying to implement these cutting edge technologies as they're developed, but we're also thinking of ways we could put them together, because each technology has a con and a pro. And we don't like to think of either or. We like to think of how can we put two things together to capitalize on their strengths and cancel out their weaknesses. And that's something that we've done. So we've put in a new technology platform, which we call ID Tracks, basically by combining several approaches for drug development that are typically used by the industry. I'm just going to name them: high content screening, target-based profiling, and machine learning. I know they're a mouthful. Doesn't matter. The point is, each one of these technologies in its own accord is an extremely powerful technology. It comes with a little bit of shortcoming, but when we put them together, we have something very, very powerful, and something that has allowed us to find answers to those questions in a very effective way. And because of implementing those technologies at the Miami project, we now have exactly the answer to those painful questions. What are gene products in the neurons that we can develop drugs towards to make efficacious drugs that will hopefully make it to the clinic and make a difference?
In doing so, you've really also emphasized the need for pharmacology, if you will, actually allowing these drugs to target different mechanisms that seem to, at the end of the day, enhance axonal regeneration better than we've seen in the past.
Right. So we just touched on the complexity of the problem of promoting axon regeneration. And I just want to take a step back and comment on something. Since the molecular biology revolution and when we first identified genes and, scientists started observing associations between mutations in a single gene and a disease, there was this assumption that for every disorder out there, for every disease, we're going to be able to find one gene or gene product that we can develop a drug towards, and that's going to resolve the disease or create a therapeutic outcome. But what we've seen over time is that that's not the rule. If anything, that's the exception, and the majority of disorders are not caused by a single mutation, and that they're complex, and that modifying one gene with a drug is not going to be enough. And this is nowhere more important than in oncology, because cancers are heterogeneous and complex, and in neuroscience applications, because the CNS is, again, complex. And in our case, as you mentioned, axon growth is repressed by multiple different mechanisms. So I'm just going to mention two. These are kind of the consensus in the field. As we age, human beings, central nervous system neurons, they lose that ability to regenerate themselves. So this is referred to as the intrinsic regenerative capacity. The other major mechanism that represses axon growth is the fact that in the central nervous system, there are molecules that signal to the axons not to grow. So this is referred to as the extrinsic mechanism because it's extrinsic to the neurons. So targeting one or the other at a time produces a little bit of regeneration, but there's, the one that you didn't target still repressing the growth. So what we found is that if you can manipulate both of these mechanisms at the same time, that's when you're getting that robust, extremely obvious regeneration of those axons. So the problem now becomes, okay, how do you target both things at the same time when it's hard enough to target one? You maybe want to try to combine two drugs, right? But as you know, developing drug combinations is very complicated, because each drug comes with its own side effects and possible negative effects. So doing this for two drugs at a time is extremely complex. So one idea that we've been spearheading is the idea of polypharmacology, where you try to develop a single molecule that does two things at the same time. And the benefit of this is that instead of having to worry about all the difficulties of developing two drugs at the same time, or a combination, you're just developing one molecule. So the feasibility of making it into a drug is a lot higher. And this one molecule can do both things. So it's going to be very efficacious. And we've so far been successful in developing this molecule, which I think is a major breakthrough and can potentially make a big difference in drug development for spinal cord injury. And we're hoping that if this success reaches the clinic, then it could open the way for using the same methodology and the same philosophy in drug development in other CNS disorders.
So in your recent lectures, we've heard about your lead compound, and you're very excited about that. Not only that compound, but other compounds you and your colleagues are working on. Do you want to tell us a little about the lead compound and where you are with that translational process?
Right. So this is the compound that I was just talking about. This compound, after we applied our technology and identified the two different targets that we need to engage at the same time with our drug candidate, we went and did a search, and we found that somebody already by chance had designed a molecule that so happens to hit those targets at the same time. But you can't have it all at once. This molecule didn't have good properties to become a drug, right? So, but it was a very good first step. It kind of opened the door for us to know that there's a starting point there. We just need to improve upon it. So we took this molecule, and it had several things that you don't want to be in a drug, without going too much into the details. But I'm just going to say, for example, it had a bad effect on the heart, right? So we needed to figure out, can we modify this molecule enough so that we keep the good things about it, the fact that it can engage two different targets that we want, promote a lot of nerve regeneration, but at the same time get rid of the negative effects. So that's what we've been doing for the last couple of years. And with the help of brilliant chemists at the Sanford Burnham Prebys Institute, we were able to engineer out those negative attributes and maintain the positive attributes. So now we're at a point where we need to continue developing this compound and make sure that it's safe to try in human beings in a clinical trial setting. But as you know, this is the hardest part, what is referred to in translational science as the valley of death, because a lot of technologies, arrive to this point, but going from this concept of a drug candidate into a clinical trial requires a lot of resources, a lot of monetary support, and this is not always easy to put together. There are mechanisms, there's government mechanisms that we're going for. We're also talking to some potential philanthropic investors and so on and so forth. But as far as the science is right now very well laid out. We're just trying to line up the resources so that we can go from where we are now to the point where we have a candidate that we can go to the FDA and say, look at our data package. This is something very promising. We're going to try that on people.
Very, very exciting, the progress that you're making. And you're exactly right. We get to a stage in academia where certain funding allows you to be successful in moving the science forward. But then all of a sudden you're trying to take it to the next level in terms of clinical trials. And that's where it's extremely expensive, trying to understand the requirements of the FDA, for example, to actually look at the pharmacological properties of the drug, the safety of the drug, all those things that have to come first. And that's why, just to emphasize your interactions with industry. I mean,
Right.
Yes, we're at the University of Miami, but you and your colleagues and others throughout the medical school are doing more and more work in alliance with industry to actually help us move our discoveries forward.
Right, absolutely. And I mean, there's almost a misconception about this because interacting with industry and interacting with foundations, and here I have to emphasize the important role for foundations, is not optional. It's absolutely necessary, because if you don't have an asset that someone is going to look at it and say, okay, that makes sense to me to make a drug out of it and take it to the market, it's never going to reach the patients. So we are focused on, delivering the value to the patients, but we have to make sure that we have the path to do that. And clinical trials are expensive. No single institution, academic or foundation typically has that kind of resource to move those things forward. So at some point or another, you're going to have to partner with a pharma company or do some fundraising to be able to push those studies forward.
Yes, and we hope that with the Miami Project's help, we can get those funds and continue to push that forward, because again, that's the business we're in, to try to move new therapies to people and see if we can improve quality of life and function. I should, before we close, just emphasize to the listeners that yes, we're talking about neuroscience today, but briefly you mentioned cancer, and also you work heavily in the kidney disease arena. So the technologies that you're developing is not just helping specifically spinal cord injury, but also branching out into other areas. And again, that's so important because we can learn from these disciplines and use new technologies and approach, and we can help other disciplines as well.
Absolutely, and that's the beauty of the Miami Project and the institutions that are, you know, close to us. This collaborativeness, because as you mentioned, a lot of these technologies are platform in nature, which means you can plug them into a separate application and you can get the same amount of scientific advance, right? So once you've invested into creating this engine, if you may, it's very easy to just port it into a different disease application. So because the Miami Project brings in collaborators from a lot of different disciplines and fields, you can really capitalize on that value. So you make the investment to develop the technology. Now our focus is spinal cord injury, but now the technology is there. So we have been trying to apply it in other applications so that we can deliver that value immediately. You're not repeating the investment. You have one investment, but you're getting a lot of return on that investment very easily. I can mention also that we're trying to develop this technology further by taking it out of the university and making a commercial platform out of it, so that also pharmaceutical companies could end up using it in the future, because we really think it could make a difference.
Well, thank you, Hassan. This has been very exciting to talk to you, and congratulations to you and your colleagues on all the progress you're making. I will have you back so that you can update us on the progress you made in terms of the next stage in this developmental platform that sounds so exciting. I really believe making a big difference in people's lives. So congratulations. Thank you very much for spending some time with us today.
Thank you, Dalton, for having me.